In this video, we are going to start talking about the reactions that have to do with monosaccharides. Now, when we have a monosaccharide, understand that under different conditions, they can be reduced to what are called aldetols. Aldetols are actually another word for sugar alcohols. And as you can see here, we have beta D glucose pyrenoids. Now, even though we know that the glucopyranose is going to be the form which is the most favorable, understand that there's going to be an equilibrium between the closed form and the open form. Once the open form occurs, understand that under reducing conditions, so for example, if we react them with, for example, hydrogenation conditions, or as you can see, sodium borohydride, you are going to be converting the aldehyde functionality to an alcohol. Okay. You may be asking, what is the use of taking a monosaccharide and reducing it? Well, for example, if you have glucose, which is also known as dextrose, because that's the sugar in the blood, once it gets reduced to the primary alcohol, that is known as glucitol, in other words, sorbitol. And that is a common sugar substitute. So sugar alcohols are going to be utilized as sugar substitutes. So many sugar-free gums contain these compounds in order to not have a sugar present that can be metabolized in our system and used for energy, but you can still have the taste of sweetness. Selective oxidation can also occur for an aldose, specifically not a ketone. And so we can utilize different chemical tests to differentiate between aldoses and ketoses. Among the chemical tests that can happen, the most common ones is the Tollens test and the Fehling's test. Understand that in the Tollens test, what is happening overall is that, once again, those glucopyranoses, or the pyranoses that come from aldohexoses, they can undergo an open form, and then the aldehyde functionality that is present in the molecule will be oxidized all the way to the carboxylic acid. In the case of the Tollens test, what is occurring is that this is called the silver test reaction. And the reason why it's called the silver test is because the aldehyde functionality gets converted into specifically the carboxylic acid, and then understand that you're going to precipitate silver, and that's why we see the second test tube illustrating a positive test for Tollens. Now, another oxidation reaction that can occur is specifically the one that is present in the Fehling's test. Conceptually, a Fehling's test is very similar to Tollens, in which we are going to be converting the aldehyde and oxidizing it to the carboxylic acid. The difference is that specifically for the Fehling test, what we have is that the complex that is utilized in the Fehling's test is going to have a copper 2 ion. And the copper 2 ion is going to undergo a reduction. We're going to go from copper 2 to copper 1. What results from that is that copper 1 oxide is going to create this compound that is reddish. Okay? And understand that it is important to know that the Fehling's test, any carbohydrate that it is positive, it is known So a positive result on the Fellings test is known as a reducing sugar. Now, it is known as a reducing sugar because the sugar is undergoing oxidation while it is having the ability to reduce the copper 2 to copper 1. So understand that reducing sugar does not mean that the sugar is being reduced itself. As you can see from the reaction, the sugar is being oxidized. Its aldehyde functionality is going to the carboxylic acid. What is getting reduced is the copper compound. So understand that glycosides, specifically sugars that have a committed anomeric position, where now we have an acetal instead of a hemiacetal, they are not going to be able to undergo oxidation. That's why they are known as non-reducing sugars. Now we are going to talk about a different type of compound, which is known as a disaccharide. As you can see in the first bullet point, the disaccharide consists of 2 sugar units, and it is connected to one another through a glycosidic bond. Now, this glycosidic bond, depending on the sugar that we're going to talk about, is going to occur between different units of the 2 monosaccharides. For example, in this slide, we have maltose. Now, maltose specifically, as you can see, is going to be, and I'm going to highlight it, a 1,4 glycoside. What that means is that maltose, which is specifically glucose linked with glucose at position 1, at position 4, they are linked through a glycosidic bond. Now, this is known as an alpha glycoside. So if you remember the little acronym that I illustrated in several videos before, Buddha, the alpha comes from the idea that at position 1, you can see that the OH is pointing down. So that's where the alpha glycoside comes from. Now, the reason why maltose is specifically a reducing sugar, so for example, if you put maltose in the Fellings test and it is known as a reducing sugar, is because we have a hemiacetal at the anomeric position. So any disaccharide that has a hemiacetal at its anomeric position is going to be able to undergo reduction in the reaction that we explained previously, called the Fellings test. The second disaccharide that we are going to talk about is lactose. When it comes to lactose, understand that lactose is formed by galactose and glucose coming together. Specifically, one of the things that we can see is that between galactose and glucose, they are going to have a 1,4, I'm going to do it in red, a 1,4 glycosidic bond between the 2 monosaccharides, but in this case we have a beta glycoside. The reason why we have a beta glycoside, if we remember the Buddha acronym, is because our carbon 1, the bond is pointing up. The next disaccharide that we're going to talk about specifically is sucrose. Now, sucrose, which is known as table sugar, understand that this one specifically is going to be a 1,2 glycoside. Sucrose, understand that can be converted into fructose and glucose, and that's what actually we have in honey. Now, one of the things that we should notice is that in sucrose, we have the anomeric positions of glucose and fructose, which are the two monosaccharides that make sucrose, compromise into a link. So the glycosidic bond that exists in sucrose is going to link both anomeric positions between the glucose and the fructose. So because the anomeric positions are making that acetal bond, for that reason, sucrose, it is known as a non-reducing sugar. The next topic that we are going to talk about is polysaccharides. And polysaccharides are going to be molecules in which we have many units of sugar. Specifically, when it comes to the polysaccharides, we are just going to mention a few of them, starting with cellulose. So cellulose is a polysaccharide that contains between 7,000 to 12,000 glucose units connected to a beta glycosidic bond. The beta glycosidic bond that is happening in cellulose, understand that it is beta 1,4 glycosidic bond. Now, cellulose strands strongly H bond with one another. Cellulose is basically the fiber that is found in many plants. Now, understand that cellulose is what's giving wood and plant material that rigid structure. Understand that when it comes to this polysaccharide, specifically that beta 1,4 linkage between all the glucose molecules that are present in cellulose, it is something that us as humans, we do not have enzymes to break them down. That's why when we consume fiber, what it does is that it actually pushes things out of the body because it is not processed. And again, it's because we do not have biomolecules, specifically enzymes, which are a type of protein. We're going to see those later. We don't have the enzymes that have the ability to break these molecules down because of the beta 1,4 linkages between the glucose. Now, one polysaccharide that we do have the biomolecules in order to break them is starch. Understand that starch is another polysaccharide that is composed of glucose units, but in this case, it's connected by glycosidic bonds that are alpha 1,4. Now, understand that when it comes to starch, there are actually two polymers of starch that are present. Starch is made out of amylose and amylopectin. Amylose is a polysaccharide that forms like a helix structure. So when we actually observe amylose, we think about it as a helix structure. Okay? That the glucose molecules are connected alpha 1,4. When it comes to amylopectin, you have that strand of alpha 1,4 glucose molecules. But one of the things that is observed in amylopectin is that it has branching. And that branching happens between the anomeric position and carbon 6 of glucose. So not only we have the alpha 1,4, we also have the alpha 1,6. In the body, we have biomolecules that are able to take starches and break them down into glucose. That's why starch is a form of energy for us. Moreover, there is a molecule that is called glycogen, and it is also known as animal starch, because one of the biomolecules that we are able to store is glucose, and we do it in the form of glycogen. Now, The glycosidic bonds that constitute glycogen, understand, that are very similar of those to amylopectin. And you can see it here where this is amylose. The drawing in the bottom represent amylopectin. And when we look and compare glycogen to amylose and amylopectin, understand that it is more closely structurally related to amylopectin, but it has more branching. Okay? The reason why it is animal starch, as I just mentioned, is because that's how we store sugar.